Hello everyone, welcome to the channel Microbial World. In this video, we will be learning about eukaryotic transcription process. Eukaryotic transcription is the process by which a segment of DNA is copied into RNA by the enzyme RNA polymerase II in the nucleus of eukaryotic cells. So in eukaryotic transcription, the genetic information in DNA is copied into RNA inside the nucleus with the help of the enzyme RNA polymerase. This RNA can later be used to make proteins or perform other functions in the cell. Key features of the process includes, as we already said, the process occurs in the nucleus. It involves multiple RNA polymerases, that is RNA polymerase 1, 2 and 3. It requires transcription factors for initiation. The process produces a pre-mRNA which undergoes post-transcriptional modifications before becoming a mature mRNA. In eukaryotes, there are three main types of RNA polymerases, each responsible for transcribing different types of genes. RNA polymerase 1, which is located in the nucleolus, transcribes ribosomal RNAs, specifically 28S, 18S and 5.8S rRNAs. RNA polymerase 2, located in the nucleoplasm, transcribes pre-mRNA, small nuclear RNAs and microRNAs. RNA polymerase 3, which is also located in the nucleoplasm, transcribes transfer RNA, 5S ribosomal RNA, and other small RNAs like small nuclear RNAs. Initiation. Initiation is the first and most complex step of transcription in eukaryotes. It involves recognition of the promoter region, assembly of transcription factors in RNA polymerase 2, and the unwinding of DNA to allow RNA synthesis to begin. Promoter recognition. Promoter is a DNA sequence located upstream of the transcription start site. The major elements of the promoter region includes Tata box. Tata box is located 25 to 30 base pairs upstream, that is before the transcription start site. It acts as a landing site for the transcription factor called TBP, Tata binding protein. Another element is BRE. The full form of BRE is TF2B recognition element. It helps the transcription factor TF2B bind to the promoter and in turn helps RNA polymerase to start transcription. INR is initiator element. It is located exactly at the transcription start site, that is +1 position. DPE is called downstream promoter element, which is located downstream, that is after the transcription start site, usually around +28 to +32. Binding of transcription factors and assembly of pre-initiation complex. Now, what are these transcription factors? Transcription factors are special proteins that helps to control the process of transcription. They work by binding to specific DNA sequences like in the promoter region. General transcription factors are needed for the transcription of all genes. In simple words, we could say that the transcription factors act like managers. They decide when, where and how much of a gene should be used. They will guide the RNA polymerase to the right place on the DNA and helps to start or stop transcription. TF2, that is transcription factor 2, refers to RNA polymerase 2. TF2 factors are general transcription factors that is needed to help RNA polymerase 2 to start the transcription process. Now let us look into the step-by-step formation of pre-initiation complex. The first step is the binding of transcription factor 2D to the Tata box. TF2D is a multi-subunit complex that includes TBP, Tata binding protein, and TAF, TBP associated factors. Tata binding protein directly binds to the Tata box. TAF stabilizes the TBP DNA interaction. In the next step, transcription factor 2A and transcription factor 2B binds. Transcription factor 2A stabilizes TF2D DNA interaction, whereas transcription factor 2B helps position RNA polymerase II at the right place on the DNA and recruit it to the complex. In the next step, RNA polymerase II arrives escorted by transcription factor 2F. TF2F helps RNA polymerase II to bind accurately to the DNA and prevent non-specific binding. Then transcription factor 2E and transcription factor 2H will bind to the complex. The function of TF2E is to recruit and regulate TF2H. TF2H is critical because it has helicase activity and kinase activity. Using the helicase activity, TF2H will unwind the DNA at transcription start site. And using its kinase activity, it phosphorylates the C-terminal domain of RNA polymerase II. So now the formation of pre-initiation complex is complete. DNA unwinding and phosphorylation of RNA polymerase II. As we have already said, transcription factor 2H helicase unwinds a small region of the DNA, almost 10 to 15 base pairs, to expose the template strand. This will create a transcription bubble where the RNA synthesis will begin. We also know that it is transcription factor 2H kinase that is responsible for phosphorylating the C-terminal domain of RNA polymerase II. Now, what is this C-terminal domain? C-terminal domain is a special tail-like extension that is found at the end of the largest subunit of RNA polymerase II. The C-terminal domain is made up of multiple repeats of a specific 7 amino acid sequence called the heptapeptide repeat. This amino acid sequence consists of amino acids such as tyrosine, serine, proline and threonine. So using the kinase activity of transcription factor 2H, it phosphorylates the serine 5 residues of C-terminal domain. Once phosphorylated, RNA polymerase II will begin synthesizing RNA. It is released from the promoter and begins to transcribe the DNA. RNA polymerase II will move forward along the DNA template to elongate the RNA transcript. Elongation is the stage in transcription where RNA polymerase II moves along the DNA template strand to synthesize a complementary RNA strand. This phase begins after promoter clearance and the phosphorylation of C-terminal domain of RNA polymerase II. RNA polymerase II breaks away from the pre-initiation complex and begins the synthesis of RNA in 5' to 3' direction. It uses the DNA template strand which it reads in 3' to 5'. Ribonucleotides are added one by one based on complementary base pairing. Adenine pairs with uracil, thymine pairs with adenine, guanine pairs with cytosine and cytosine pairs with guanine. Shortly after initiation, RNA polymerase II pauses downstream of the transcription start site. This is called promoter proximal pausing. This pause is stabilized by factors such as negative elongation factor and DRB sensitivity inducing factor which binds to RNA polymerase II. This pausing allows proper capping of mRNA and recruitment of elongation factors. The post-transcriptional modifications will be discussed in detail in the upcoming videos. If RNA polymerase II has to continue the elongation process, PTEFB is recruited. PTEFB stands for Positive Transcription Elongation Factor B. PTEFB phosphorylates serine 2 of the C-terminal domain. This phosphorylation recruits RNA processing factors for capping, splicing and polyadenylation. Transcription factor 2S plays a role in rescuing backtracked polymerase II. If RNA polymerase II encounters a block and backtracks, the 3' end of the RNA moves out of the active site, halting the transcription process. Transcription factor 2S is a rescue factor which is like a helper that comes in when RNA polymerase II is stuck. It stimulates cleavage of the nascent RNA, realigning the 3' end back into the active site. This helps the transcription process to restart and continue the elongation process efficiently. Termination is the final phase of transcription where RNA polymerase stops RNA synthesis, releases the RNA transcript and dissociates from the DNA template. In eukaryotes, especially for RNA polymerase II, this process is very complex and coupled with RNA processing. The first step in the termination process is the transcription of polyadenylation signal. As RNA polymerase II elongates the pre-mRNA, it transcribes a polyadenylation signal, 5-AAU-AAA3'. It is located 10 to 35 nucleotides upstream of the cleavage site. A protein complex including CPSF and CSTF binds to the polyadenylation signal. CPSF is cleavage and polyadenylation specificity factor. CSTF is cleavage stimulation factor. This binds to the polyadenylation signal and cleaves the nascent RNA strand downstream of the AAU-AAA site. After cleavage, the next step is polyadenylation. Poly-A polymerase adds a poly-A tail which is about 200 adenines to the 3' end of the RNA. This step is essential for RNA stability, export from the nucleus, and translation initiation. In the next step, an exonuclease enzyme XRN2 binds. XRN2 stands for 5-3' exoribonuclease 2. The exonuclease enzyme XRN2 binds to the free 5' end and degrades the RNA rapidly. Once XRN2 catches up to polymerase II, it dislodges or destabilizes the polymerase and RNA polymerase II is released from the template, leading to termination. After termination, RNA polymerase II is dephosphorylated and it is recycled for another round of transcription. So this is all about eukaryotic transcription process. I hope that the video was useful for everyone. Do subscribe the channel for more informative videos. Thank you.